Imagine a world where we can rewrite the genetic code of life, design organisms from scratch, or even create entirely new species. Sounds like science fiction? Not at all. We are on the threshold of a great era, the age of synthetic biology, where laboratories are not just places of research, but also places of creation. Are you curious about the endless possibilities and ethical challenges it brings? Join me to discover. Welcome to the Hidden Past channel with the series 70,000 Years of Human Secrets. In this video, we will explore the topic, the age of synthetic biology, creating life in the lab. Don't forget to subscribe to the channel so you don't miss other videos exploring the mysteries of humanity. If you find this video interesting and useful, please like and share to support us. Synthetic biology is a promising interdisciplinary field that combines engineering principles and biology to design and construct new biological components, devices, and systems. It's not just about modifying genes, but creating biological functions that never existed before, or redesigning existing biological systems in completely new ways, much like engineers building a machine. The main goal is to transform biology into a predictable engineering discipline, enabling the design and assembly of biological systems with specific functions and broad applications. Unlike traditional genetic engineering, which primarily focuses on moving or altering specific genes, synthetic biology has a broader vision. It doesn't just edit genetic code, but aims to create new biological software, including entire DNA strands, genomes, or even complete cells and organisms designed from scratch. The core difference lies in the level of control and the scale of intervention. This field envisions a future where scientists can program life at will, creating organisms capable of performing complex tasks that no natural organism can. From bacteria producing clean fuels, plants that glow, to immune cells that kill cancer, the potential of synthetic biology is limitless. It opens a new era of biological innovation where the line between natural and artificial increasingly blurs. Synthetic biology is not just a collection of tools or techniques, but a new philosophy of how we interact with life. It is based on the idea that biological components can be standardized and assembled like electronic components. This allows for the construction of complex biological systems in a more systematic and efficient way, accelerating the pace of discovery and application. The principles of design, build, test, and learn are central. The foundation of synthetic biology is built upon a deep understanding of genes, proteins, and metabolic pathways, combined with breakthrough advances in DNA sequencing and synthesis technology. The ability to accurately read and write genetic code has turned bold ideas into reality. We are moving from merely understanding how life works to actively designing and shaping it, bringing both unprecedented opportunities and enormous responsibilities. The history of synthetic biology, though a relatively young field, originates from fundamental discoveries in molecular biology and genetic engineering from the mid-20th century. Early breakthroughs in understanding DNA structure and genetic code laid a solid groundwork. Recombinant DNA technology in the 1970s, which allowed gene transfer between organisms, is considered foundational for the ability to design life. However, synthetic biology only truly emerged as a distinct field in the early 21st century as DNA synthesis technologies became faster, cheaper, and more accurate. A significant milestone was in 2000, when W. French Anderson's team created a simple genetic circuit in E. coli cells, expressing fluorescent proteins in a defined pattern. This is considered the earliest example of applying engineering principles to build biological systems from standardized components. One of the most prominent figures is J. Craig Venter. In 2010, his team successfully announced the creation of a bacterial cell with a chemically synthesized genome named Synthia. This was the first time a living organism was created from an artificial genome, opening the door for designing and building organisms from scratch. This event caused a sensation, sparking many debates about the definition of life and ethics. Since then, the field has seen rapid development of new tools and techniques. CRISPR-Cas9 gene editing technology, introduced in 2012, has revolutionized the ability to precisely edit genetic code, becoming an indispensable tool. CRISPR allows DNA to be cut and pasted with unprecedented accuracy, significantly expanding the scope and complexity of biological systems that can be designed. The advent of programs and competitions like iGEM also plays an important role in promoting synthetic biology. iGEM encourages students to design and build biological systems using biobricks, creating a vibrant community of research and innovation. These events help disseminate knowledge and create a repository of reusable components and methods. These milestones have shaped synthetic biology into a rapidly developing scientific and engineering field, with the potential to completely transform how we produce everything, from medicine to fuel. Continuous progress in the ability to read, write, and edit genetic code is bringing us closer to transforming biology into an engineering language, where we can program life for specific purposes. To realize the ambitions of synthetic biology, scientists have developed many core technologies. One of the most important pillars is DNA synthesis. The ability to create custom DNA sequences is fundamental to rewriting or designing new genomes. Initially slow and expensive, advances in automation and chemistry have made it faster, cheaper, and more accurate, allowing for the synthesis of longer, more complex DNA sequences. Besides DNA synthesis, gene editing technology has become an indispensable tool. In particular, the CRISPR-Cas9 system has revolutionized the field. CRISPR allows specific genes to be targeted and edited with high precision and efficiency. It acts like molecular scissors that can cut DNA, allowing sections of genes to be added, removed, or changed, opening new doors for designing complex genetic circuits. Another technology is large-scale genetic engineering, which includes creating large plasmids or artificial chromosomes containing multiple genes or entire metabolic pathways. This allows for the assembly of large biological modules to perform complex functions, far beyond the capability of editing a single gene. Technologies like DNA assembly allow multiple small DNA fragments to be joined into larger genetic structures. Modular design and standardized components, biobricks, are also core principles. The idea is to build a library of interchangeable biological components, each with a defined function, and then assemble them into more complex genetic circuits. This approach is similar to using standard electronic components to build devices, making the process of designing biological systems more systematic and repeatable. Detection and measurement technologies are also important for evaluating performance. Techniques such as RNA sequencing, mass spectrometry, and biosensor systems allow real-time monitoring of gene expression, protein production, and metabolic pathways. This provides the necessary feedback to optimize design and troubleshoot problems. Finally, the development of computational tools in biological design has been instrumental. Gene design software, biochemical network simulations, and artificial intelligence are helping to predict the behavior of engineered biological systems and optimize components before experimental implementation. These tools significantly reduce time and cost, accelerating the design, build, test, learn cycle. In medicine, synthetic biology is opening unprecedented doors for diagnosing, treating, and preventing diseases.
One of the most prominent applications is the production of drugs and vaccines. Bacteria and yeast are being redesigned to produce complex compounds, such as insulin for diabetes, artemisinin, an effective anti-malarial drug, or morphine. This helps reduce production costs and increase access to medicines. Additionally, synthetic biology is also used to develop new, more effective vaccines. Instead of using weakened pathogens, scientists can design specific proteins or DNA sequences to stimulate an immune response, resulting in safer and easier to produce vaccines. mRNA vaccine technology, although not entirely synthetic biology, shares many similar design principles, demonstrating the potential to program biological responses. Another promising area is gene therapy and cell therapy. T immune cells can be redesigned to recognize and destroy cancer cells more accurately, as in CAR-T therapy. Stem cells are also being studied to repair damaged tissue or provide new functions. Synthetic biology allows for the creation of smart cells, capable of sensing pathological environments and releasing drugs when needed. Disease diagnosis is also an area where synthetic biology is making breakthroughs. Biosensors designed from bacteria or proteins can detect early signs of cancer, infections, or harmful substances. For example, bacteria can be engineered to change color when exposed to specific carcinogens, providing a fast, low-cost diagnostic method. Creating beneficial gut bacteria, genetically modified to combat diseases, is another direction. These bacteria can produce anti-inflammatory or immune-boosting compounds, helping to treat digestive or autoimmune diseases. The ability to intervene in the human microbiome with high precision opens a new era of personalized medicine. However, with this potential come significant challenges in safety, ethics, and regulation.
Introducing genetically modified organisms or cells into the human body requires rigorous testing and strict monitoring. But it cannot be denied that synthetic biology is shaping the future of medicine, offering hope to millions suffering from severe illnesses. Synthetic biology promises a green revolution in energy and environment, providing sustainable solutions to global challenges. In energy, the focus is on developing next-generation biofuels. Scientists are designing bacteria, yeast, and algae to directly produce ethanol, butanol, or gasoline and diesel-like hydrocarbons from biomass or CO2. This can reduce reliance on fossil fuels and carbon emissions. Furthermore, synthetic biology also aims to optimize photosynthetic efficiency in plants or microalgae to produce energy more effectively. By redesigning metabolic pathways, these organisms can convert solar energy into valuable fuels or chemicals with higher yields than in nature. A typical example is the design of bacteria capable of producing hydrogen, a clean energy source. In environmental protection, synthetic biology offers great hope for cleaning up pollution and waste management. Bacteria have been engineered to perform bioremediation, i.e., to break down hazardous pollutants such as oil spills, pesticides, or heavy metals. For example, specific bacterial strains can eat plastic, addressing the global plastic waste problem. Another potential application is the ability to sequester carbon dioxide, CO2, from the atmosphere. Photosynthetic microorganisms can be designed to absorb CO2 more efficiently, then convert it into useful materials or fuels, helping to mitigate the greenhouse effect. This provides a biological approach to combating climate change. Synthetic biology also contributes to the creation of sustainable materials. Instead of using toxic chemicals, engineered organisms can produce biodegradable biomaterials, such as bioplastics or other polymers.
This helps reduce non-biodegradable waste and negative environmental impacts, creating a more circular economy. Environmental monitoring systems are also improved by synthetic biology. Sensitive biosensors can detect the presence of pollutants at low concentrations, helping to monitor water, air, and soil quality continuously and accurately. The ability to program life to solve environmental problems is a huge step forward, paving the way for a more sustainable future for the planet. In agriculture, synthetic biology has the potential to revolutionize how we produce food, address food security issues, and reduce environmental impact. Scientists are designing crops that are more resilient to drought, pests, and harsh soil conditions. This helps maintain crop yields in the face of climate change and dwindling resources. By editing genes or introducing new genes into crops, we can enhance their nutritional value. For example, creating golden rice rich in vitamin A is an achievement, and synthetic biology continues to expand this capability, creating crops that produce essential vitamins, minerals, or proteins to combat malnutrition. The goal is to develop crops that not only provide energy, but also complete nutritional sources. Another exciting application is the design of soil microorganisms to improve plant health and reduce the need for chemical fertilizers. Nitrogen-fixing bacteria can be genetically modified to work more efficiently, helping plants absorb nitrogen from the air themselves, significantly reducing the use of polluting nitrogen fertilizers. Similarly, microorganisms can be designed to combat plant pathogens. In materials science, synthetic biology is opening a new era of high-performance and sustainable biomaterials. Scientists are designing bacteria or yeast to produce proteins with unique properties, such as spider silk protein. Spider silk is renowned for its superior strength and elasticity, and mass production of this type of material could create super light, super strong materials for many applications. Additionally, synthetic biology is also used to develop self-healing materials.
Microorganisms can be integrated into materials to produce healing agents when the material is damaged, extending product lifespan and reducing the need for replacement. This can be applied to building materials or fabrics. Creating materials that can self-repair like biological systems is a major breakthrough. Designing organisms to produce bioplastics is also a key focus. Instead of relying on petroleum, bacteria can be programmed to convert sugars or agricultural waste into biodegradable plastics. This not only solves the plastic waste problem, but also creates a more sustainable production process, reducing the burden on the environment. Beyond these great promises, the age of synthetic biology also poses countless serious ethical, social, and legal challenges. One of the biggest concerns is the issue of playing God. Creating life in the laboratory, designing organisms that never existed, or profoundly altering the nature of natural life causes many to worry that humans are crossing ethical boundaries and interfering with sacred processes. The line between creation and science becomes blurred. Another concern is the risk of biosafety and biosecurity. If engineered organisms escape into the environment, they could cause unforeseen consequences for ecosystems, compete with native species, or cause new diseases. The ability to create engineered pathogens or biological weapons is also a serious threat. Strict control over technologies and materials is crucial to prevent misuse. Intellectual property is also a complex challenge. Who owns an organism entirely designed in a laboratory? Can genes, biological circuits, or even entire genomes be patented? This affects access to life-saving technologies and could create monopolies and inequalities. Balancing encouraging innovation and ensuring public benefit is a difficult task. Additionally, there are deep questions about equity and access. Will the benefits of synthetic biology be distributed fairly globally, or will they be concentrated in wealthy nations and large corporations? If only a few have access,
It could deepen social stratification and create new biological divides. The challenge of legal regulation is also immense. Current regulations are often not designed to handle the complexity and speed of development.
Flexible and comprehensive legal frameworks are needed to manage the research, development, and application of synthetic organisms, ensuring safety, accountability, and transparency. Achieving international consensus on ethical standards and regulations is also a difficult task. Finally, synthetic biology also raises philosophical questions about the definition of life, human nature, and our place in it. If we can create life from scratch, does that diminish the value of natural life? Do these artificial organisms have any rights? These questions have no easy answers and require the participation of scientists, ethicists, philosophers, policymakers, and the public. The future of synthetic biology is a vibrant and incredibly promising picture, far beyond what we imagine today. We're moving towards an era where designing biological systems will become as easy and predictable as writing computer software. Tools and techniques will continue to improve, allowing us to work with increasingly complex biological systems, from single cells to cellular networks and even artificial tissues and organs. One of the most exciting prospects is the ability to personalize synthetic biology. Imagine a future where medicines are produced on demand for each individual based on their specific genome and health needs, or crops designed to grow optimally in specific conditions. Personalized medicine and precision agriculture will be completely reshaped by the ability to program life. The development of living machines or biocomputers is also a promising direction. Cells can be designed to perform complex calculations, store information, or act as tiny sensors and actuators. This could lead to smart implantable medical devices, autonomous environmental sensor networks, or even new forms of artificial intelligence. The biggest challenge in the future remains building appropriate regulatory and ethical frameworks to keep pace with the speed of innovation.
Continuous dialogue among scientists, policymakers, ethicists, and the public is needed to ensure that synthetic biology is developed responsibly, benefiting all of society without causing irreversible risks. Educating the public about this field is also extremely important. The ultimate goal of synthetic biology may be the ability to create life entirely from scratch, with a completely artificial genome capable of performing complex functions on demand. This is not only a scientific achievement, but also a leap in our understanding of the nature of life. However, achieving this goal requires not only technical skill, but also deep wisdom regarding the limits and responsibilities of creative power. The age of synthetic biology is opening new horizons, where the boundary between natural and artificial life is increasingly blurring. We are slowly grasping the power to change and create life, a capability once only belonging to creation. This not only leads to breakthroughs in medicine, energy, or the environment, but also raises deeper questions about the definition of life and death. And when we can design life, can we also design immortality? This is a big question that we will explore together in the next video. Immortal humans. How close is science getting? Don't miss it.